Hello everyone. In this video, I will talk about the SIP database that we have developed in our lab, which is stress combination and their interaction in plants database. So I will focus in this video, I will focus on the Phenomics dataset, which has been collected and curated for various plant species under three major categories of combined stress, which is abiotic, 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 biotic, and biotic, biotic. Firstly, let's just go over a quick introduction of what combined stress in plant means. So when a plant is growing in the environment, there are several environmental or abiotic factors such as light, water, and temperature that can affect plant growth. Also, there are other biotic factor or microorganisms such as bacteria, fungus, virus, and nematode that can induce disease conditions in a healthy growing plant. Thus, these abiotic or biotic factors can affect individually or can have a simultaneous effect on growth of the plant. So any imbalance in one of these factors will result in, in an unhealthy plant growth. So there is a need to understand this interrelationship between plant and its various environmental factors. So why do we need the combined stress database? So firstly, combined stress studies are being conducted by various scientific groups all over the world. However, this information still remains scattered. By developing a platform like SIP database, we are simply trying to connect the dots and develop a knowledge base and to understand what is available and what is not available. In future, a strategy can be devised which can be used for crop improvement. So this SIP database will basically act as a repository of information on combined stress studies and provide information on the effect of stress combination on plants in a simple way. So what does SCIPDb offers? So SCIPDb will have information regarding various host pathogen interaction and plant rate information, distribution of studies conducted globally through the transcriptomic data sets. Identification of potential target genes for crop improvement can be identified. We have collected and curated like data for various cultivars which are there in the original research articles, and response to erratic climate changes can also be understood through our SIP database. So looking at the publication trend on combined stress articles, we see that we have observed that number of papers are increasing every year. So here also you see there's an increasing trend. Every decade there is an increase in number of articles which are being published on combined stress studies. And because of the volume, the the SIP database has literature. The literature has been collected and curated till December 2021, and because of the voluminous size of the available literature, and since the collection and curation is done manually, it is highly challenging and demanding. So also it requires a qualified manpower. However, the update is still ongoing because the article and the volume of the data available is too much. So the work is still ongoing till December 2021. Let us now go to the database live and see how can we uh, scroll through the SCIPDb and to understand the Phenomics dataset. So as already discussed in the previous video about the structure of the database, I will go through the Phenomics section of the database and will give a tutorial on how the SCIPDb database can be used to understand the Phenomics dataset. So this is the home page view, and the tabs are located in the top header area. On the right side of the home page, you see on the right side of the data page, you see the number of the literature which has been hosted, the number of stress combination, the number of organisms, and the transcriptome and trait which has been listed in this database so far. So if you click on any of this link, you will be transferred to the PDF downloadable file, which can be downloaded by the user to understand, to get the list of what is there in the database. If you click on the organism, you will get the list of the organism. The user can scroll through the list of what has been hosted so far in the database. To understand how the methodology what is the methodology followed for the collection and curation of the database, you can access it through the About tab. So the user can simply go to the About tab and click on the methodology link. So the methodology link will redirect you to this page. This is a flow chart of methodology which has been followed so far to collect and curate the data. So broadly, the literature mining was done from various search engines. Then the articles were segregated into research articles and ancillary articles. The segregation of research article was done on various morphophysiological data sets, which has been categorized as parameter A, B, and C, and which has been used to extract values, analyze the data into negative and positive. So if the user is interested to go to understand the detail of the methodology, it can be easily downloaded from this link, which will redirect the user to a PDF downloadable file. And here the user can scroll through how the literature mining was done, what are the search engines which we have used, how the articles were sorted into ancillary and main research articles, listing of the parameters, how it was done. So broadly there we have categorized parameters as type A, B, and C. So type A parameter includes the growth and yield related parameters such as plant height, biomass, seed weight, seed number, seed test weights, etc. So these type A parameters, they directly reflect the impact of the stress. The type B parameter includes physiological parameters such as photosynthesis, stomatal conductance, transpiration, chlorophyll content, etc. And also the disease parameters such as pathogen load, disease score, etc. So these type B parameters, they indirectly reflect the impact of the stress. And lastly, the type C parameters, which include the biochemical parameters such as proline content, MDA content, nutrient content, ROS content, etc. They also explain the impact of the stress, but to a lesser extent compared to the other two classes of parameters. Scrolling down the same file, the user can understand how the calculation of the data was done and how the data was analyzed and interpretation of the data. And finally, this data was integrated in the database. So for new researchers and for research people who are not aware of combined stress terminologies, they can click here for the various terminologies, list of plant species and organisms which have been included in this database. So by clicking on this, there is again a PDF downloadable file which the user can download to understand what has been included in this database. Now let's go back to the home page. So on the right hand side, there are two major data sets which have been hosted in our SCIPDb. So one is the Phenomics data set and the other one is the transcriptomic data sets. So in this current video, I will talk about Phenomics data sets. So let's click on Phenomics. So Phenomics is an important aspect of the database because to make sense of other omics data like transcriptomics and proteomics, it is important to have an understanding of the Phenomics data set. So when you click on the Phenomic data set, this page will appear. So this has combined stress Phenomics data for various simultaneous and sequential stress. So if a user wants to go to so the question is how the user will navigate the data which is hosted in our database. So there's a brief description which is given on the right hand side. So the user can go through this description to understand what exactly is there in this Phenomics section. So here's a view more tab. So if you will click here, it will expand this description. So here it has been explained what simultaneous and sequential data sets exactly mean with respect to the combined stress studies. So now let's just take an example to understand how this Phenomics data set, Phenomics section works. So first we will choose a category. For example, simultaneous stress abiotic biotic. In the second category, the user will be allowed to choose which of the stress combination he or she wants to look at. So let's take flooding and high temperature. And then after choosing a stress combination, the user will have a list of plants on which the studies have been collected and curated so far. So here in this particular stress combination, studies have been conducted on 5 plants. So let's select cotton and submit. So this page will appear on the screen. So this is the data page which has been generated for combined flooding and waterlogging and high temperature stress for the plant cotton. So on the top, there will be a heading indicating which stress combination is this. Then there will be a brief introduction about this stress combination. Moving, if you scroll down, then there will be the name of the plant. Then the data pages for individual research articles will follow. So this is for one study and the other studies will follow up after one study has ended. So this is study 1 and this is study 2 and following study 3. So in total, this cotton plant has 4 studies listed in the data page. So broadly, a data page will have the study number, the net impact, the outline of uh, the outline of the data page, which will have all the information like crop, which crop was used in this particular study, the stress, name of the stress 1 and name of the stress 2, stage of the plant which was used. Secondly, a tabular data will be there. So this tabular data will have the details about the data which is there in the original article. If you scroll down, then there will be a note uh, which will show how the calculation was done from the original data itself to obtain this value on the tabular, which is represented on the table. Then there will be a interactive scroll bar and then a summary, which will summarize one particular study pertaining to one particular research article. So this particular study, so all the studies in the database will have two major links. One will be the raw data link. So if a user will click on this, a downloadable Excel file will be downloaded. And if you will click on this, so this will help user download the original raw file or processed data. So this is the same data which has been reflected in the tabular form here. The second link will be the reference link. So this if the user will click on this reference, then the user will be redirected to the original research article. Also, we have one feature in the database. So to understand the dynamics of the net impact on the tabular data when different degrees of stress have been tested. So we have this scroll bar, which if you will drag on the available stress concentration will show you an overall impact on the plant. For example, here, if you see the tabular data has two variable values for the waterlogging stress. So if you will scroll, so currently the scroll bar is at 34-degree temperature and 3 days of waterlogging. The net impact is green, which is not affected under combined stress. So if the user will drag it to 6 days of waterloggings, so then the outcome changes and becomes red in color, which says that it is more affected under combined stress. So now let's go back to the Phenomics page. So this example was related to abiotic. Now let's go take another example from the biotic category. So this time let's select sequential biotic category. And let's select this stress combination as flooding and insect. And when you will open this third drop down menu, you will see that this stress combination has been reported so far in 4 plan. Let's select maize and click submit. Here also, when the data page will open for maize plant, so there on top there will be the name of the stress combination, which is flooding, waterlogging, submergence and insect stress, a brief description about this stress combination. If you'll scroll down, the name of the plant will be there. On the right hand side, there's an outline which will summarize which is the crop which has been studied in this study, and what is stress 1, what is stress 2, and stage of the plant. So here in this particular example, there is the stress 2 is a biotic factor. So in such condition, the data which is represented in tabular format is divided into two major categories. First category is the net impact of the individual and combined stress on plant growth, and the second category is the interaction between stress 1 and stress 2. So here the interaction between flooding and insect is represented under combined stress at plant interface. So here the user will understand like how stress 1 and stress 2 are impacting each other when they are affecting the plant. Similarly, as described earlier, if the user is interested to download the processed or the raw data file in an Excel format, it can be downloaded through this link. So when the file is downloaded, you can open it. To see the detailed Excel file. Also, the reference link will redirect the user to the original article. And at the end of the data page, summary for the article has been provided to give an understanding of the data presented. Let's go back to the Phenomics page and look another example from the third category, which is biotic. Let's select simultaneous stress, biotic. And under the subcategory 2, let's select fungus, nematode and fungus as stress combination. So when you select the third drop down menu, you see that this stress combination has been reported in several important crops. Let's select pigeon pea. Submit. So here again the data page will appear for this crop, pigeon pea or Cajanus cajan. So here the user can scroll down to see how many studies have been listed so far. So this is number 1. This is and other studies. This is the type study. Then this is the following study 1. This is 2, 3. So in total 4 studies have been listed for this particular uh, crop. So the generic pattern again here would be the outline of the individual study, which will indicate the crop, stress 1, stress 2, and the stage of the plant. The tabular data, which will reflect the net impact of individual and combined stress on plant growth, following which there are 3 links here. So apart from raw data and reference, which have been already explained earlier, the third link here is the genotype study link, which is a downloadable PDF file. So in original articles where information regarding other genotypes have also been included, that information has also been included in our database. So if the user is interested to understand how the other genotypes of this plant are affected by this particular stress combination, they can download the file from this link, and this file can be downloaded. So this is the description telling how the tabular data was calculated. The interactive bar showing overall impact of stress 1 and stress 2 on the crop which is not affected. The interaction between nematode and fungal pathogen under combined stress. So this will show the interaction of stress 1 and stress 2 at the plant interface, and the brief summary of this particular study. So coming back to the phenome page, if you see here, top left of the page has a link to visualize the overall available data in the database. So click here to reach the various visualization which have been provided to understand the volume of the available data on SCIPDb. So the first visualization is the bubble chart. So if the user will click here, so an interactive bubble chart will appear on the screen. So this is only for abiotic studies. So here if you see, these are bubbles of various color and sizes. So the size of the bubble indicate the number of study, and different color represents the different stress combinations here. For example, for drought and heat here, these many crops have been reported in our SIP database, and the maximum number of studies have been reported for Arabidopsis. So if you move the cursor on the bubble, so it will reflect the details about that individual bubble. Coming back to the visualization page, similar bubble chart is available for the abiotic stress combination. So if you click here, the user will be redirected to the bubble chart for abiotic stress combination. Here again, different studies, stress combinations are represented by different color codes, and the size of the bubble represents the number of studies which have been included. Going back to the main visualization page, The third one is the bubble chart for biotic-biotic stress combination. So this reflects the number of studies which have been hosted so far under this category. And the major data set is coming from the nematode fungus stress combination, which indicates that it is an extensively studied stress combination so far. And these are the crops which have been reported in this particular stress combination. So another way of visualizing the data available in the SCIPDb is the stress matrix. So if the user will click on stress matrix link, this chart will appear, this matrix chart will appear, which is available for biotic-biotic stress combination, a separate page for abiotic-biotic stress combination, and for the abiotic-abiotic stress combination. So overall, a user can visualize how much data is available in the SCIPDb, and it has been color coded as red and green and blue, which overall indicates the impact of this stress combination on the plant. For example, drought and heat is coded in red color, which means that the combined stress is more deleterious in Arabidopsis. So it has been coded as red. However, the stress combinations in which the combined stress is not as deleterious as the individual stress has been color coded as green. And there is also a third category in which the combined stress has been coded as blue in color. So here in these particular studies, the conclusion was not the studies were not conclusive and definitive to say whether it is negative or positive. That is why we have included in the others category. So there are various types of filters which are available. So if the user wants to filter the information based on various parameters, so it can be filtered out using biomass. So all the stress combinations which have biomass as one of their parameters will be filtered out. Like this, there are various parameters which can be used to filter out the information from the stress matrix. User can also filter out, let's mark it to all. So the second type of filter which is available is the plant category. So if a user is interested to only look at alpha alpha, or for example Arabidopsis only, then it can be filtered out. Then all the studies for Arabidopsis will be filtered out. So like this, we can filter out the studies for Arabidopsis only. And the third category of filter is this filter by stress combination. So here the user can filter out one specific stress combination or more than one stress combination. Like if I want to filter out drought and heat and drought and highlight, it can be filtered out like this. So the next category of visualization which is available on the SCIP database is the radial tree depiction. So the fifth category is a radial tree depiction of biomass. The user will click on this. So the percent reduction of biomass under individual and combined stress is available for abiotic stress. So here the user will be shown the data for abiotic stress combination and related crops, and at the end of the radial chart there's a bar diagram which represents the degree of change. So this radial chart is available for photosynthetic rate also, stomatal conductance, chlorophyll content, disease damage. So the last visualization is the geographical distribution. So these are the studies which have been included in the database. So there's a description on the left hand side. So a user can go through this description to understand what these location pins which have been shown in red, green and blue color means what is the color code used for these description. And these locations can be filtered out by three broad categories, which is abiotic, 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 biotic, and biotic, biotic. So let's only see what abiotic stress category has. So if we filter out abiotic here, so the shown pins are showing these pins show the studies which have been conducted under under this category. So if the user will click on this pin, so the information regarding one specific pin is reflected. So this pin reflects salt and UV stress combination, which was conducted in Karachi, Pakistan, and it was conducted in the year 2020, 2012. And here the outcome is summarized. So which is shoot length was more reduced under combined stress, under higher stress level. And this is the location of the study. Likewise, if we click on green pin, so one is, so this will show description about this stress combination. So this is wounding and drought, which was conducted in Mexico in the year 2015, and here increased lignification under combined stress was observed. So like this is an user interactive map which will tell about all the studies which have been included so far in the SIP database. Now coming back to the main phenome page, there is a tab for metaphenome. So let's click on this. So this is again an interactive representation of the net impact of stress combinations on plant performance. So under this section, we have listed stress 1, stress 2 and stress 3 and which plant species a user wants to look at. For example, let's select salt and heat. So if the user will select stress 1, stress 2 and plant species, so this kind of data is reflected. So this is red in color, which shows that combined stress of salt and heat is more deleterious compared to the individual stress. A representative graph has been shown here showing that combined stress has more mortality rate compared to the individual stress. Let's take a different example here. Let's take drought, bacteria, rice. So here the color is green, which indicates that the combined stress is less deleterious compared to the individual stress. So here the description is given as reduced blight lesion under combined stress was observed. Let's take another example of flooding, insect, broccoli. So here the color is gray. So the gray color here depicts that the outcome from this particular study was not conclusive as red or green. So that is why the gray color has been given. So to conclude, what SCIPDb Phenomics section offers is that a user can analyze the extent of coverage of an individual stress combination in various plant species. It provides a holistic view of available literature, helping to locate the voids or gaps in combined stress understanding. An area of focus can be assessed for future research ventures. Also, this is an attempt to address few important questions which are listed in the MPMI Harris et al. Paper, and SCIPDb can be used to understand how does abiotic stress such as climate change influence plant microbe interaction, and how do microbe interaction affect plant microbe interaction. For more detailed application of SCIPDb, please go through the application video. I would like to acknowledge NIPGR and SERB India for providing the funding for our project. Thank you for listening.